I'm going to be presenting some research from my dissertation work with Tom Parchman at the University of Nevada, Reno, on the landscape genomics of a foundational plant in the western United States. So hopefully this will load. So broadly what I'm interested in is looking at how geographic space and environmental variation influence genetic variation and maintain genetic variation through both demographic and adaptive processes, and thinking about the types of experiments and analyses we can use to tease those two things apart. Okay. Oh, there we go, that's fine. Cool, so yeah, geographic space, environment, blah, blah, blah. All right, so as populations begin to move across the landscape, experience different demographic changes, say something like population expansion, they experience new and novel ecological and environmental factors, and are influenced by things like genetic drift and gene flow, and with that they're influenced by environmental variation that can have divergent selection and cause divergent selection in a few different ways. Local adaptation from direct selection from environmental or ecological processes, isolation by adaptation or niche partitioning where two populations occupy different niches within the same geographic space, or environmental variation can have dispersal barriers that limit gene flow. So we can use what we know about outcomes and expectations from these processes to assess how demographic history and adaptation confer the landscape genetic variation that we see today, and essentially look back in time. So the study system I'm going to be using is rubber rabbitbrush, or Ericomeria nauseosa. It's a foundational shrub species that exists in a wide variety of environmental factors or regions from Mexico to Canada, and is really wildly phenotypically diverse with over 22 named subspecies or varieties. It's also due to recent anthropogenic change like fire and overgrazing, and the introduction of invasive cheatgrass is one of the many plant focal species for restoration purposes throughout the Great Basin. It's also wind pollinated. Oh, that's fine. It's upside down. So it's wind pollinated and also is the only flowers in the early fall. So it's the only thing that available for many insects to feed, nectar to feed on. And given all these reasons, its ecological importance and all the different environmental variation and experiences, along with the phenotypic diversity, I'm going to argue it's an ideal system for trying to understand mechanisms that mediate genetic variation in really complex western landscapes. It could be informative for restoration in other desert communities. So our overall objective here is to link geography and phenotypic variation with geography, environmental variation, and evolutionary history. We're going to do that by asking 3 questions. 1, how is genetic variation structured across the landscape? 2, what are potential, how does environmental variation shape genetic variation across space? And what are the contributions of geography, environment, and genetics to phenotypic variation? Of course, the 3 different types of data we're going to be using are genomic, environmental, and phenotypic. Here you see our 17 sample populations from across the Great Basin. We used a genotype by sequencing method called ddRADSeq from 222 individuals and ended up with around 39,000 SNPs. So we analyzed spatial genetic structure and did some environmental associations in a few ways. We used ordination techniques like PCA and RDA. We estimated population genetic parameters like for genetic differentiation and diversity, and we also used both packaged and custom Bayesian models to link geography and environment to genetic and phenotypic variation. I don't really get into very many specifics about genomic pipelines or the analyses, but feel free to ask me later. The environmental data we use is this climatic water deficit toolbox. It includes 30 different climatic variables that have been shown to influence spatial variation in plant communities. And we also have lat long and elevation. So So if you focus in on some of these things, while they don't look that different, they actually occupy a wide range of temperatures and precipitation and a huge elevation gradient. We have phenotypic data from a greenhouse common garden experiment with 100 plants per population for our 17 populations. This is part of a larger collaborative at UNR with Beth Ledger's group and a PhD student, Allison. They're using field and greenhouse common garden experiments for native restoration in a wide variety of plants throughout the Great Basin. So from this, we get 59 highly detailed root and biomass traits that have been shown to influence establishment and early survival in early stages of development. And we also have survival estimates from 40 and 60 days. So first, we're going to look at genetic variation. And we see really tight population structure. What you see here is a Procrustes rotation of a principal components analysis in lat long. So what this does is it takes those two matrices, kind of rotates them together, and either stretch or constrains them. It gives you some sort of estimate of how correlated they are, and that's the R squared you see down here. And pretty much the points here are exactly the same color as the map, and they pretty much look the same. And also, if you focus in, this is Reno, Nevada, where I live. These are all within 10 kilometers of my house. I can actually walk my dog, take my house and walk my dog where these were sampled. And here they are in principal component space. They are really tight, non-overlapping clusters here in just a really short geographic space. Next, we're going to look at environmental variation in a couple ways. First, we're going to look at the overall contributions of geography and environmental distance. We're going to look at population-specific drivers of environmental variation and look at specific environmental associations. So here are correlations between genetic distance and geography and environment. So you get estimates of isolation by distance and isolation by environment. So why these correlations are important, they don't really get at how geography and environment drive genetic partitioning or genetic structuring, and they're not really, they're hard to compare the two in effect size. So what we did was use this program Bedazzle. What it is a Bayesian, hierarchical Bayesian model that it tries to quantify the extent in which allele frequency shifts are explained by geography and environment. And we found that geographic distance roughly explains 3 times the amount of the genetic structure among populations as environmental distance. Next, we showed that our 16 of our environmental variables seem to be driving genetic partitioning we see among individuals and populations. So these, using a partial redundancy analysis, so these can be kind of busy to look at, but the magnitude of the lines you see here is the strength of the correlation to a point, and the rotation of a point to a line is the relationship of that correlation. So here, for example, these individuals down here are strongly negatively correlated with maximum winter temperature, so these individuals all have a lower maximum winter temperature. Furthermore, you can use RDAs to kind of identify outlier loci or candidate loci that might be associated with particular environmental variables. And we ended up finding 391 associated loci. And most of these seem to be associated with temperature and precipitation in the fall and winter, which you would kind of expect in this plant that flowers in the fall and seeds in the early winter. Next, we look at phenotypic variation. And I admit I'm kind of new at building these hierarchical Bayesian models, but it's something I'm really interested in exploring. And I'm going to just talk through what I've been thinking about what I've been doing with these. And so I used distance matrices of genetics, environment, and geography to explain 2 different phenotypes. I used seedling emergence and a composite variable of our root traits that have been shown to influence establishment. So the figure you see down here is the post-year outputs from that model, and you can, in terms of beta coefficient or the effect size, and all 3 seem to have a pretty large effect on seedling emergence. And if you're more familiar with a p-value type analysis, the 95% credible intervals around these do not overlap 0, so you can say that they have a p-value of less than 0.05. I think really technically this 95% credible interval does overlap 0, but all we really care about here is the effect size and how these jointly contribute to that. And we did the same thing for root traits, and we show that genetic variation is the only thing that seems to be affecting root traits. which you would expect since all these things were reared in common gardening experiments, so we removed the effect of environment and geography. And I've been kind of thinking about this as I got here, and I think this does make sense, kind of. So seedling establishment seems to be dependent upon genetic variation in the environment in which the seed was produced in. By including geography as a covariate in this model, it kind of removes any neutral population effects maybe, and there might be some G by E interaction here that is indicative of an adaptive response to seedling emergence. And obviously here it just means that there's population-based genetic differences in root trait variation. So with that, in conclusion, genetic variation, we find that there is really strong population structuring that seems to almost perfectly mirror geography. While geography seems to be explaining a lot of the variation we see in the genetic structuring, we do find there is population-specific environmental drivers that is partitioning genetic structuring, and maybe have some candidate loci that are associated with environmental variation. Here we find that geography and environment variation, along with genetic distance, are jointly explaining this, and we find genetically based population differences for root trait. I think this is the most important result down here, and something we're going to be interested in exploring more and playing with, especially with the Ledger Lab, because if we're able to predict seedling emergence or seedling establishment or different root traits that are important for establishment, just based upon the environment in which you exist in or the amount of genetic variation you have, this can have really important impacts for restoration. And with that, I'd like to acknowledge the Carson Lab, the Ledger Lab, especially Allison for growing and rearing all these things, and people helping me build some of these models. Also, if you're interested in learning more about this and the potential for reproductively isolated lineages of rubber outerbrush, an undergrad in our lab is presenting a poster tomorrow on this. And with that, I'll take any questions or comments.
Do you know anything about, so is there subtle variation in flowering time amongst these subpopulations?
Yeah, there is. So we just have anecdotal evidence, so we're kind of just getting into all this. So last year, or a year and a half ago, we went out and sampled 1 million different plants and looked at it. We didn't really expect to find that much variation between these different subvarieties, but there's like anecdotal evidence that there's different flowering times in reproductive isolated lineage, stuff like that, within the same geographic space.